Good morning, everyone. I am Tanesh from Ransit University, Thailand. May I introduce the next presentation? The title is Screening of Single-Chain Variable Fragment.
Everyone here?
Everyone hear me?
Loudly?
Okay. Again, yes. Okay. Good
Okay.
morning, everyone. Here. Can you hear me?
Yes.
Okay.
Yes. Okay. Okay. May I introduce the next presentation? The title is Screening of Single-Chain Variable Fragment specific to express PD-L1 by Cholangiocarcinoma KKU213 cell line for cancer immunotherapy. The presenter is Mr. Methi Wathikthinnakon from Chiang Mai University. Dr. Methi, Mr. Methi, are you ready?
Yes.
Okay. The moderator, please prepare the slide for Mr. Methi. And
Okay, just minutes, please.
Do you hear me?
Wait a minute.
wait for the slide presentation.
Okay, thank you.
Wait for a while.
Okay. Everyone can see?
Yes, I can see.
Okay.
If
Okay.
Mr. Methi ready, please welcome Mr. Methi
Ready.
to start the slide presentation.
Yes. I can't control. Okay. Yes. Good morning to all the audience. My name is Methi Wathikthinnakon. I am a PhD student in Department of Biology, Faculty of Science, Chiang Mai University. This morning, I would like to present about the screening of single-chain variable fragment or ScFv specific to express PD-L1 by cholangiocarcinoma KKU213 cell line for cancer immunotherapy. In this talk, I am going to give you some information about cancer and cancer immunotherapy, and also the interaction between PD-L1 and PD-1. The objectives and research workflow will be presented in the middle of presentation, following with the result and discussion. Finally, the conclusion will show in the end. The first topic I would like to talk is a bit about cancer. As we know, the cancer is the disease that caused from abnormal cell growth. The abnormal cell can invade its originated organ and has ability to spread throughout the body and finally cause death to the patient. The risk factors of cancer are many. It can be result from the mistake in DNA level until the environmental level. Cancer is the second cause of death worldwide, and it is the first cause of death in Thailand for many past years. The awfulness of cancer comes from the fact that many cancers can be detected in patients only in the late stage. Even successful surgery, the cancer recurrence rate is still high and survival rate is low, although the common adjuvant therapy like chemotherapy or radiotherapy has been used. Cancer seems to respond more aggressive. In these few decades, there is the rise of the cancer immunotherapy, since it has been reported that cancer immunotherapy can give more effective results in both in vitro and in vivo. Cancer immunotherapy is the promising curative approach, since it is the use of immune system to fight cancer by targeting immune cells, especially T cell or NK cells, to the desired cancer. In this picture, you can see that there were many approaches to bring T cell to kill cancer. So if we expect the effective of using of cancer immunotherapy, it is important to make T cell ready and fully function. On the left picture show the expected scene when the receptor on T cell surface bind to an antigen on cancer cell. T cell will be activated and kill cancer cell. Unfortunately, some cancer have an antigen named PD-L1 on their surface that can bind to the receptor named PD-1 on T cell. It's shown in the picture on the right. The interaction between PD-L1 and PD-1 resulting in T cell exhaustion that make T cell not function, and the cancer can escape from immune surveillance. There were several studies reported that administration of inhibitor of PD-L1 PD-1 interaction could significantly increase the efficiency of immunotherapy. This suggests that the combination of PD-1, PD-L1 inhibitors and other immunotherapy treatments would be beneficial to enhance immune cell function. Thus, the present study aims to screen human ScFv specific to PD-L1 for enhancing immunotherapy. ScFv protein that neutralize PD-1, PD-L1 interaction can be used in various platforms of immunotherapy. So, the objectives of this study is to screen anti-PD-L1 ScFv from human ScFv phage display named YAMO library and determine the binding specificity by flow cytometry, and to examine the diversity of the obtained ScFv by PCR-IFLP, DNA sequencing, reduced amino acid alignment, and protein structure remodeling with Swiss model web server. Here is the workflow of this study that can be divided into 4 parts. First, the biopanning was performed to screen the human ScFv phage display from YAMO library. The biophages that can bound to PD-L1, recombinant of PD-L1 was the biophage were eluted and transfected into E. coli HB2151 stain. The growing clones were identified for the positive clone by colony PCR. The positive clone DNA fingerprints were demonstrated by PCR-IFLP technique. After protein expression, the culture supernatants were subjected to binding specificity tests using flow cytometry. The top 5 clones were cultured and extracted for the plasmid DNA that contain ScFv genes and used in DNA sequencing. And finally, the DNA sequence of the top 5 clones were analyzed for reduced amino acid sequence using IMGT request online software, and protein structure were remodeled by using Swiss model web server. Here is the result. We got 12 positive clones from transfection of E. coli 2151 with eluted biophage. Then PCR-IFLP was performed, and DNA fingerprint shows that here that there were 11 unique clones, since the clone number 9 and number 32 has the same fingerprint. The ScFv from each clone were expected to be secreted in the culture supernatant after protein expression. So the culture supernatants were used in binding specificity tests. The treatment of the experiment were stain the cell with indirect staining method, like here. The top 5 clones were shown on the bar graph in the blue color, with the positive control demonstrating that KKU213 that we used expressed 99.5% PD-L1. The amino acid sequence alignment of VH from each selected clone is shown here. All anti-PD-L1 ScFv from these 5 clones consisted of 4 framework, like here, and 3 CDR in the color of yellow. The critical differences amino acid sequence were found on CDR2-VH and CDR3-VH domain, since CDR regions provide the recognition site of antibody and ScFv. So the variation between the sequence, especially in CDR2-VH and CDR3-VH domain, might result the anti-PD-L1 ScFv from the selected clone to bind different epitope or maybe have a different binding affinity to the same epitope. To demonstrate the protein structure, the amino acid sequence of 5 clones were submitted to Swiss model web server for homology remodeling. The results show that all 5 anti-PD-L1 ScFv structures consisted of VH and VL domain linked by linker peptide that shows in this picture, and all of them have high similarity to compare ScFv in Swiss model database. CDR2VH and CDR3VH that have high differences among them were colored with the green and yellow respectively. However, the correlation of protein sequence and the structure and binding affinity in addition to neutralizing activity inhibit PD-L1 PD-1 interaction still need to be further evaluated, and the potential anti-PD-L1 ScFv will be further used for development of immunotherapy in future. For the conclusion, after biopanning, we got 11 unique clones, and only 5 clones were selected due to their binding specificity. After analysis of DNA sequence, amino acid sequence alignment, and protein structure, prediction of 5 selected clones show that they have a potential to use further in evaluation and maybe used in further immunotherapy study in our lab. So finally, I want to acknowledge my advisor, Dr. Usra Panya, for being the guide and the supervisor of my work. And I would like to say thank you to all of this in this list for the opportunity and grant to do this research. Okay, thank you.
Okay. I'm sorry about the internet option fail. Can you hear me?
Yes.
Okay. Thank you for Methi.
Thank you for your presentation,
Okay.
Methi. Okay, and because the time's out, if you have any question from the audience, please type in the chat box, and I think Dr. Methi will answer you again.